Have you ever thought about what you're made of? No, I don't mean stardust, although that's true too. I mean the tiny, tiny building blocks that make up every part of you. Your skin, your hair, your heart, your brain, everything. These are called cells. Cells are like microscopic cities bustling with activity. Each cell is a self-contained world: power plants, factories, transportation systems. It's a universe in miniature, working tirelessly to keep you alive and kicking. This is where our story really begins. Get ready to zoom in and explore the inner workings of this cellular metropolis. All right, let's meet the stars of our show, the GERL System. The name GERL is an acronym, which is a fancy word for a name made from the first letters of other words. It stands for Golgi, endoplasmic reticulum, lysosomes. Think of them as a super efficient delivery and maintenance crew inside your cellular cities. Each member has a specific job, but they work together like a perfectly coordinated relay race. It's teamwork that makes the dream work, even at a microscopic level. First up, the endoplasmic reticulum, the ER. The ER is like a massive factory complex with long tunnels and workshops. The Golgi, the post office, lysosomes, the recycling center. Understanding how this team works is fundamental to understanding life itself. Let's go. We're touring the GERL system. Let's take a closer look at the endoplasmic reticulum. The ER is a vast network of membranes, folded sheets, and interconnected tubes. It stretches throughout the cytoplasm and connects directly to the nucleus, the cell's control center. The nucleus holds the DNA, which sends instructions that the ER uses to start building. The ER comes in two types: the rough ER and the smooth ER. The rough ER is studded with ribosomes, the protein-making machines. New proteins enter the ER lumen where they fold into their correct shapes. Getting the shape right is critical. A misfolded protein won't work, like a key cut wrong. The smooth ER, without ribosomes, makes lipids and helps detoxify drugs and poisons in cells like liver cells. Once proteins and lipids are ready, the ER pinches off transport vesicles, tiny cargo bubbles. These vesicles protect their contents as they travel through the busy cytoplasm toward the Golgi. Their primary destination? The Golgi apparatus. The ER has done its job. Now it passes the baton to the next team member. Next stop, the Golgi, the cell's sorting and shipping center. Let's see how it works. The transport vesicles filled with proteins and lipids butt off from the ER and head to the Golgi. They travel through the cytoplasm to our next stop, the Golgi apparatus. The Golgi looks like a stack of flattened, pancake-like sacs called cisternae. Think of it as a stack of pita breads. Vesicles arrive at the receiving, or CIS face. They fuse and empty their cargo into the Golgi lumen. As cargo moves through the stack, special enzymes modify and refine it. The Golgi might add sugar molecules, glycosylation, or trim and tag proteins to help them fold and find their destination. It's the gift wrapping and addressing stage. Finished products arrive at the transface and are packed into new vesicles. The Golgi creates different types of vesicles for different destinations. Some fuse with the plasma membrane to release contents outside. Others go to internal organelles. Some Golgi-derived vesicles mature into lysosomes. Without the Golgi, proteins would float around aimlessly. The Golgi brings order to the chaos. It's the ultimate cellular logistician. Next, the lysosomes, the cleanup crew. Now we come to the final member of our GERL team, the lysosomes. These are the cell's cleanup and recycling crew. Some Golgi vesicles are filled with a cocktail of powerful digestive enzymes. They are little bags of acid and enzymes, safely contained by a strong membrane. They break down waste materials and worn-out cell parts. They also destroy invading bacteria and viruses. When an organelle is old, the cell engulfs it and fuses it with a lysosome, autophagy. Lysosomal enzymes break it down into amino acids and sugars that the cell can reuse. Lysosomes help defend the cell. When a white blood cell engulfs a bacterium, a lysosome fuses with that vesicle and destroys the intruder. The ER builds, the Golgi processes and ships, and lysosomes clean and recycle. It's a continuous dynamic cycle that keeps the cell healthy. This coordination ensures the cell has resources, responds to its environment, and stays functional. It's a beautiful, efficient, essential dance of molecules. Science. Thanks for joining the tour of the GERL system. Explore more. Try a microscope lab or a 3D cell model activity.